let's now turn attention to individual decision making, particularly the decisions involving simple choices and the application of the drift diffusion model to these simple choices. But before we get started, I want to just read out this quote from Fehr and Rangel in the paper that I'm recommending to read in the literature down here. And what it's saying is basically that the brain controls human behavior. Obviously, we know this from neuroscience, behavioral neuroscience in particular. And economic choice is no exception to this. So why am I stating this here? Well, it's basically to remind you that we're trying to understand how the brain controls economic choice. But let's turn our attention to simple choice. And this is the example that I'm giving here is from a very common experiment in this domain of research. So we're giving participants the chance or the opportunity to choose between two different food items. Let's say a Twix bar and a bag of chips here on the right. Now, in most of these studies, these participants are relatively hungry, so they've been through 4 to 6 hours of fasting before they show up to the lab, to the experiment, which means they're hungry, they're motivated to make the right choices. And they also typically have to stay about 30 minutes to 60 minutes in the lab where they then get to consume the food that they chose over. So while they're already hungry, they even have to wait a little bit longer. And the only food they get is the food they choose in the experiment. So the stakes are pretty high for these participants. And the experiments are basically about choices between a small number of familiar goods. For instance, like choosing between apple and orange for dessert. There's no uncertainty involved. That's the important point here. It's just about picking one out of two food items, and then one trial typically gets rewarded at the end. Now, this type of research has led to a model that was proposed by Fehr and Rangel in a 2011 paper, which is part of the recommended reading. And this model has 5 key components, which we'll discuss in separate videos. So the first one is that the brain computes a decision value for each option. And the important thing here for neuroimaging is at the time of choice. So this is when the participant is considering their choice options. The brain also computes an experience utility signal at the time of consumption, which is at a later time. So once you've chosen something, this is typically what's scanned, you then get to experience what you've chosen. And that's the experience utility signal. The third point is choices are made by comparing decision values using the drift diffusion model. So this is one way that we can model decisions, and we'll go over the drift diffusion model in a separate video. Finally, decision values are computed by integrating information about attributes. So each choice item has attributes. Is it red? Is it sweet? Is it salty? Those kinds of things that are associated with each option and their attractiveness. So we'll talk about attributes and how they may be integrated in a model. And the final point is computation and comparison of decision values are modulated by attention. So attention can influence how this integration occurs that eventually then leads to decision values, the integration of information regarding attributes. So we'll talk about these 5 points in detail now, but let's get started with the first key component of this model. And that's that the brain computes a decision value for each option at the time of choice. So just to remind you, what we mean by decision values or action values, those are the signals that are computed at the time of choice. And they basically are a way to estimate or predict the, well, in a way, hedonic impact of choosing from different options. So you can estimate how good you're going to feel about consuming each one of the different choice options, which is not saying that you're basically estimating subjective value at this time point. And then supposedly you're picking the item that gives you the highest subjective value. The hypothesis here is that these signals exist and causally drive choices. and they do, as I'll show you in a minute. And this is supported, obviously, by systematically replicated findings, and we'll discuss some of these studies in a second. A key and candidate area within the brain is the ventromedial prefrontal cortex, or also the medial orbitofrontal cortex, and I'll tell you a little bit about this in a second. So this is the system that's involved in processing rewards or positive effects of decisions during the time of choice. So we have the ventromedial prefrontal cortex here, but it's also referred to as the medial orbitofrontal cortex in some papers. And then we have the ventral striatum, which is also an important region in this. So the typical empirical approach to study this is we give hungry participants, a choice between different food items. We first let them rank those food items in terms of their preference, because we have to make sure that we understand each participant's preferences, which can differ from subject to subject. Then we can estimate the stimulus value from behavioral data, which could be the bits during the experiments or the rankings before the experiment started, based on the choices, right, that participants make in the experiments. We can use behavioral models to estimate this if you want to use a model-based approach. But you can also just use, for instance, the willingness to pay on each trial. That is also a perfectly fine parameter that gives you an estimate of the subject's willingness to pay for this item, the subject's value for this item. While participants are performing these experiments, we can measure their neural activity. We can use fMRI for this, and obviously the common approach is to use a whole brain scan while participants are performing these experiments. And then finally, we can use statistical methods to test whether estimated signals modulate neural activity. So this is an approach that's commonly referred to if we use a model-based approach, or if we use an approach that estimates the value the subject puts on each item, on each trial. Then we can use what's called a parametric modulator to predict the magnitude of the signal. I'll have a note on this in a second, but first a sort of note on neuroanatomy conventions. So in modern papers, we refer to the region that's involved in these types of decisions. So this region right here in this meta-analysis, the ventromedial prefrontal cortex, we refer to this as the ventromedial prefrontal cortex. However, if you read other papers, they might refer to some regions as the medial orbitofrontal cortex. There's a lot of overlap between these two regions. The neuroanatomical name for this region, in fact, is the orbitofrontal lobe. If you look at an atlas, for instance, this will be typically referred to as the orbitofrontal cortex here. And so this is how, for instance, older papers and monkey papers refer to this. However, given that it's actually a larger area that also moves outside of the OFC, so the orbitofrontal cortex, more recent papers in neuroimaging refer to this as the ventromedial prefrontal cortex. So don't get confused by different naming conventions. This is something that persists in neuroanatomy. Let's now consider this initial study on neurocorrelates of basically decision values in the medial orbitofrontal cortex. So what the authors did here is they asked their participants to fast for 4 hours before the experiment. And then participants went into the scanner and went through 100 bidding trials. And the bidding trials were for food items like this. So they only saw one food item. This is not a binary choice. It's just a single item they see. And then they're asked to give a bid, which could be anywhere between, I believe, $0 and $3. And then waited. And at the end of the trial, one of their bids was actually implemented, and they had a half hour to eat the price, basically. If they actually chose to bid for it. If they didn't choose to bid for it, then they wouldn't get anything, but they also wouldn't lose the money associated with it. So these are free bid trials, and those are obviously the important aspects, but there were also forced bid trials in which the subject saw the item, but then the basically computer algorithm determined how much the subject had to bid, and then the subject just entered the number. That's all that happened here. So there's no free choice. This choice is forced. So the price is predetermined, and at the end the subject had to pay the price anyway. So we have free bid trials and forced bid trials. And just as a reminder, in neuroimaging, we always have to have a good control condition. So when we look at activation, it's typically always relative. So we have a experimental condition that we're interested in that contains all of the elements we're interested in, and we have a control condition that contains everything except for the aspect that we want to study. So we want to study free bits in this case. Namely, we want to make the subject to make a decision at this point and consider how much do I value this, which the subject in this control condition cannot do because the bid is predetermined. So there's no real decision aspect there. But visually, it's all the same across these two conditions. In terms of the movements that the subject has to make and the motor activations, they should also be controlled for, as they will also be the same. So we're controlling for everything except the element of choice, and that's what they're interested in here. You can also see the timeline here, which is something that you commonly see in neuroimaging papers because timing is important, and to understand the timing that the experiment is used allows you to see whether there may be some flaws in the experimental design. Remember, the hemodynamic response is a bit sluggish, so we have to adjust for this fact when we have multiple subparts of a trial in our experimental design. So let's move on to the results. So they find an activation in the medial orbitofrontal cortex, but only in the free bid trials, not in the forced bid trials. And the nice thing about their design is that it allows them to look at the value that they're giving here during the bid trial. So you can see that it's pretty flat here. So the orbitofrontal cortex does not have anything to do with making these forced bid decisions in quotation marks, because they're not decisions in that sense. You can see that in the free bid trials, the largest activation is during the highest bids, and the smallest activation is during the lowest bids, and then these two intermediate values also fall where they should be, right? Basically where they expected. So you have this ordering here in activation patterns, indicating that value is computed in this region here. What's not so nice maybe is that it's a relatively small activation. You wouldn't see this nowadays, but also the statistical procedures have improved. The hardware has improved. So we typically don't see such activations around just a few voxels anymore. So this is a very interesting experiment that showcases this sort of parametric effect that we have here. And in fact, you could add this into your model, and that's what's done nowadays, and also adds to some power in the experiment, which is called parametric modulation. So you can make predictions in your models now about the magnitude of the response. So let's say you have this experiment here where you basically have one volume represented on the x-axis. So here we've recorded 10 volumes, here we've recorded 80, here we've recorded 100 volumes, and you have a predicted response. In a normal model, which is shown in the red, you're just making a prediction about when a response that is involved in the task should occur. And that's already an approach that is valid and that allows us to identify regions involved in, let's say, decision making. But if you're interested in a region that tracks a specific value, such as this medial orbitofrontal cortex here, then you can also make predictions about the magnitude of this response. So let's say on this trial, the subject makes a bid of $2, on this trial of $1, on this trial of $0, and on this trial a bid of $3, right? So you can say that if we have a region in the brain that tracks value, then it should also ramp up its value when the value is actually higher, and that's reflected in the bid that the subject makes and the behavior that the subject makes. So you could have a regressor that also includes the magnitude of the subjective value to the subject and then try to identify regions that track exactly this subjective value, right? And just to give you an example, this is something that we did in a somewhat older experiment, where we had participants make decisions over lotteries that offered a gain amount and a loss amount at the same time at a 50-50% chance. So the question we asked our participants is, do you want to play this lottery or do you want to keep your money, basically, with a chance to win more money if they played the lottery, but also a chance to lose some money if they didn't. And we then summarized on each trial the amount, based on the decisions that they made, we summarized the lotteries as in the form of a subjective value, which can be computed by simply adding up the loss amount and the gain amount with some parameter that's based on the decisions that participants made throughout the experiment. And what we're finding is that in the ventral striatum and the ventromedial prefrontal cortex, there is this positive relationship between this. So between the value that a subject holds over the lottery and the activity in these two regions. You only want to look at the blue lines here. So that indicates that basically these two regions, the ventral striatum and the VMPFC, they track exactly the value, subjective value that is sort of individualized to each participant based on their decisions throughout the experiment. So they track the value throughout the experiment quite nicely, right? You can see that activity in both of these regions ramps up when value is high and decreases when value is low to the participants. So when it's actually negative, in a sense, when the losses outweigh the gains. Ignore the red line now, because that is when they make these decisions in a condition of threat, so when they're highly anxious. And it looks like, in that case, these regions no longer track this value. You can also use this information and make predictions about the choice that a participant has made based on the signal in these two regions. And you can see that in a logistic regression framework, the regions predict, so that the activity in these regions predict whether subjects are going to choose for or against playing this lottery. But let's summarize this. So basically, what has been replicated across many studies is the fact that VMPFC, and this is across different studies, activates to different aspects of decision value, of the value that of a stimulus or an item that participants see at the time point of the decision. This was also the case in this study, and this was the case in this study. So we're going to look now over a range of studies looking at these types of lotteries that we also used in our study, looking at choosing over items that you could purchase in the store otherwise, money, but also food items, and finally also donations to people in need. All of these are types of values are processed in this region here in ventromedial prefrontal cortex. And this is a meta-analysis from Rangel and Fehr. So finally, I'd like to point out that it's important that if we make such inferences, for instance, that subjective value is encoded in ventromedial prefrontal cortex, then it's preferable if we have converging evidence for such statements. So I want to also mention this lesion study because it uses a different method to basically show a similar thing. So in this study, the authors, Fellows and Fehr, and it's already an older study, they looked at VMPFC damage. So how does VMPFC damage affect choices over preferences? So we had a VMPFC damage population, so a number of participants that had specific damage, and you can see the extent in some people, but they have this overlap in VMPFC. So red means that most participants had it, but purple means that possibly only one participant had this damage. And typically when we do brain damage or lesion studies, then we also have a set of control subjects that also have some damage to cortex, but different regions in the cortex. So these are people that have lateral frontal cortical damage, but also some medial. Obviously, that's one of the problems. Typically, these types of damages are based on accidents, and so you might have more wide-ranging damage in some than others. But you can look and compare participants with specifically VMPFC damage to others that have sort of non-specific damage in other regions of the brain. And then what the authors did here, they asked their participants to perform a perceptual judgment task, which is simply, is this line longer than another? So they showed 2 lines to participants, right? And then they ask which line is longer, left or right. And this is something that control subjects can do with no damage. Well, actually, these are these controlled subjects here. But there were different types of controlled subjects that they used, right? Most importantly, however, this was not this ability was not significantly different in terms of the number of errors. They made a bit more errors in these tasks than controlled subjects, but not significantly so. So the VMPFC patients did not differ from all the other patients. Now, they did differ on another judgment task, namely a preference judgment task. And on this task, participants were asked to answer questions about what kind of food items do you like better? Do you like an apple more or an orange more? What kind of celebrities do you like better? I don't know who they used in this, but you could show pictures of celebrities and ask, do you like this one better than this one? And finally, what kind of colors do you like better? And what you find there is a large amount of intransitivity. So that means that participants said, I like an orange more than an apple, and then an apple more than a banana, but I do like a banana more than an orange. So this is an intransitivity, because if you were consistent in your choices, you should also say that you like an orange more than a banana. But it turns out that is not the case in these. So we observe these specific impairments in participants with VMPFC damage. And this is compared to IQ and age-matched controlled subjects, right? So they shouldn't be different in any other domain. But it looks like that VMPFC damage specifically impairs making preference statements or making preference judgments, which obviously agrees with what we've learned about the VMPFC in the previous neuroimaging studies, namely that it's involved in assessing subjective value, which is also obviously an important underlying computation for making these kinds of preference judgments. So this is giving converging evidence that the VMPFC is clearly involved in subjective value, in computing decision value, from which we showed from both the neuroimaging perspective as well as from a lesion study perspective. So there's converging evidence for this finding.